Welcome to Easy Microbiology.
Good morning, everyone. Today I am going to provide an overview of Clostridium difficile, including its habitat, morphology, cultural characteristics, biochemical characteristics, host range, virulence factors, mechanisms of antibiotic resistance, and epidemiology. I will be delving into each of these topics in further detail over the following slides. Let's get started. C. Difficile, also known as C. Difficile is a gram-positive, spore-forming bacterium that can cause serious infections in patients who have taken antibiotics. It is typically found in the intestines of healthy adults. However, antibiotics that disturb the equilibrium of bacteria in the gut can contribute to C. Difficile spores germinating, creating toxins that harm the intestinal lining and resulting in diarrhea, colitis, and inflammation. Clostridium difficile, commonly referred to as C. difficile, is a bacteria commonly found in the intestinal tracts of humans and animals, as well as in soil, water, and food. Its spores are highly resilient to their environment and can survive for extended periods of time, creating the potential for contamination of surfaces and objects. As such, it is essential to adhere to effective hygiene and cleaning procedures. C. difficile is a rod-shaped bacterium that forms spores, allowing it to withstand harsh environmental conditions. These spores are extremely heat and chemical disinfectant resistant, remaining in the environment for prolonged periods.
Clostridium difficile can be grown on selective agar media, such as cycloserine cefoxitin fructose agar, CCFA, which is used to identify the bacterium in clinical specimens.
Clostridium difficile is a gram-positive, anaerobic rod-shaped bacterium that doesn't require oxygen to survive and can only live in low-oxygen areas like the intestines. It can ferment various carbohydrates and releases organic acids and gases in the process. This bacterium can cause serious infections and complications and is seen more frequently in hospital settings. Thus, it is important to be aware of its presence and to properly manage it. C. difficile is a bacterium that is responsible for causing infectious diarrhea in humans, with its toxins being the primary cause. It can also lead to more serious conditions such as pseudomembranous colitis. If left untreated, it can be fatal. Spread through fecal contamination, C. difficile can infect both humans and animals, but it is most commonly found to affect humans. Thus, it is crucial to be aware of the signs and symptoms of C. difficile in order to ensure timely diagnosis and treatment for its effective management. C. difficile produces two major toxins, toxins A and B, which can damage the cells in the intestines and cause inflammation. Additionally, some strains of C. difficile produce a binary toxin, which is thought to further increase its virulence. Clostridium difficile is emerging as an antibiotic-resistant bacterium with one of the highest rates of resistance, making it critical to find new treatments and prevention strategies. Uncovering the mechanisms of antibiotic resistance is essential to prevent and combat the serious implications of this infection. C. difficile is an anaerobic bacterium that can cause significant morbidity and mortality if left untreated. It is a major cause of antibiotic-associated diarrhea and can be spread through contact with contaminated surfaces or objects. Good hand hygiene, judicious use of antibiotics, and proper cleaning and disinfecting of surfaces are critical for preventing the spread of C. difficile infections. C. difficile is a type of bacteria that can cause severe diarrhea and colitis if it overgrows in your intestinal tract. It happens when the bacteria disrupts the natural intestinal flora by releasing toxins that damage the intestinal lining and cause inflammation. If left untreated, C. difficile infections can lead to serious health issues. Regarding C. difficile, research has revealed that the production of antibodies against its toxins can give some kind of protection against potential infections. The immune response to C. Difficile infections is intricate and there is still much to be explored. When it comes to diagnosing C. difficile infections, laboratory methods such as culture, microscopy, and molecular techniques are used to detect and identify the bacteria or its toxins in clinical specimens such as stool or rectal swabs. Early diagnosis is essential in developing an effective treatment plan. Precautionary measures such as proper hand hygiene and contact precautions in healthcare settings can help prevent C. difficile infections. Additionally, antibiotic stewardship programs can be used to reduce the use of broad-spectrum antibiotics. Following these guidelines will help prevent the spread of this potentially harmful bacterium. We come to the end of our presentation, discussing the treatments of Clostridium difficile infection. This infection typically involves the use of antibiotics such as metronidazole or vancomycin to target the bacterium. Recurrence of the infection is common. However, fecal microbiota transplantation is a procedure that can be used to restore balance in the gut microbiota. Thank you for your attention and for listening to our presentation.